Hello, everybody, and welcome back. In the last video where I was talking about the number of prokaryotes, I made a goof and I talked about the quantified cell as giving us a different number of bacteria found in your gut intestines. And the paper I actually meant was the revised estimates for the number of human bacterial cells in the body. And one of the authors here is Ron Milo, the person who wrote the BioNumbers book and who also maintains the BioNumbers database, who is also an author on the quantified cell, Ron Milo. However, that was with Rob Phillips, and that's a different paper than the one I meant. We're going to read the revised estimates, I believe, the week after next. So we will get to that one. And today we're talking about the quantified estimates. I will say just very quickly that that prokaryote estimate for the number of cells inside of our intestines is wrong, right? That is a wrong number. We now have a much better number for that, and it's okay that that number is wrong because for each person that we're talking about, that's 10 to the 23rd cells, and is small compared to the total number of eukaryotes found everywhere else in the world. So I do want to point that out that number is an error. But let's talk about this quantified cell paper. This should be consistent with everything we've been understanding and reading so far. But I want to talk about this quote by Darwin, and this is in the Physical Biology of the Cell book, but they also put it here and I want to talk about it. I have deeply regretted that I did not proceed far enough at least to understand something of the great leading principles of mathematics. For men thus endowed seem to have an extra sense. So, we'll just say, look, what Darwin was onto here was that for any person, all people, right, gender or non-gendered, knowing mathematics is it's like having another sense, right? Hearing a number and going, wait, that doesn't make any sense because I know another number is a stunningly good skill to have. And it's one that you're all building now, right? If I told you that there was a protein that was a micron long, you would go, what? How is a protein the size of E. coli? Right? I mean, maybe there is, but that would be a surprise to you because you now have an anchor. You now have something that you can think about when you hear a micron and you know what that means, okay? If I told you that ribosomes were half a micron in diameter, you would say, that can't possibly be true. There are hundreds or thousands of ribosomes in every E. coli cell. And so they talk about the power of estimates, and they talk about it with this kind of neat car example. And it would be nice if cars only cost $10,000 anymore. But they talk about this car example, and they talk about making estimates that are too precise to be useful. So like, oh, how much is a Honda Civic? Well, it's $12,895 at my dealership. That's a really accurate number, right, for that dealership. But it doesn't help you understand the cost of any other car. And so they say, well, what if we say a car costs 10 grand? Is that a terrible number? And the answer is no. Like a cheap car might have been 8 grand. If I told you today a car costs 10 grand, you'd be a little surprised because inflation blew that up, right? Brand new cars I don't know what a low end brand new car is, but I know that brand new cars that I've been looking for my family, 35,000 is like pretty much the cheapest that will hold 5 adults or close to adults. So, these numbers are changed, but they give us an anchor to understand these numbers, right? So if I tell you a car costs about 10 grand, that's not terrible because it's closer to 10 grand than it is, say, 100 grand, or is it? We want to think about these things logarithmically, right? So if a true cost of a car is 40 grand, that's now more than halfway. So maybe 100 grand is a better number. There is no steadfast exact rate rule. You've got to figure this out when you're comparing these numbers. But if I want to compare different things, right, a TV costing $1,000, which is probably a little bit pricey for a television anymore, is a good way to compare those different numbers. But what I want to get to is stuff that we see later on. I want to admit, first and foremost, that the first time I was asked this question, I got it wrong. And as soon as I found out the right answer, I went, oh, right, of course. But I was asked, what what is bigger, the mRNA or the protein? And my first thought was like, I know that proteins are like these really substantial objects in cells that if you get enough of them together, I might be able to visualize them. And so I was like, oh, the protein. But I mean, it's not even close to being true, right? mRNA, which often contains coding sequences, right? And that's what you see in the orange here. For some reason, my pencil is not writing. Hello, Mr. Pencil. You're allowed to write. Maybe my Apple Pencil died. I'm sorry, everybody. I'm a noob streamer here, but we're going to figure this out. If you look carefully at the colors, you can see that there are these yellow sections that are not coding sequences. And then we've got this orange chunk that is a coding sequence. And then we have another yellow sequence that is not coding, followed by the tail. And so going from 5' to 3', we would see that we encounter this first piece, then we encounter the coding piece. Now, this may not look like it, but this is a linear polymer. What do I mean by that? I mean, if I were to pull the two ends of this, it would stretch out and be a straight line. It might look like it is connected maybe 2 or 3 times in a spot, but that's just the depiction of this thing, which is a realistic depiction of what it might look like when you're seeing it. But this really is like a long piece of string. If you pull it, it becomes straight. There is no cross bonding or side bonding or anything like that. This is a linear molecule. And then what we know is that this linear molecule is going to get translated in the ribosomes to form the proteins, right? And that translation is going to find a particular part of mRNA that's going to dock up to it, that's then going to give you the amino acid, that's then going to make it. But we're going to take 3 amino acids and turn them into 1 amino acid, right? And so that's what's happening here. And then that protein, while it is also, this is a linear molecule, meaning that if I grabbed the 2 endpoints and I stretched it out, it would become a line. Proteins are almost never encountered in a linear form. They often fold in very intricate ways that influence what their function is, okay? And so, you know, this part here is the only part that's actually the coding part. Everything else is just there to help it get done. But you've got this basically distillation of a factor of 3 down to the amino acids. And you see that dividing that by 3 is going to get you a number that's much bigger than that. And that's because there's all this other stuff that's here that's important for the decoding process, but isn't itself actually being decoded. So we take this filament that's quite long, right, of mRNA, and it compactifies down to make that one protein. But that's not the end of the story. This piece of mRNA might be read by a ribosome 10 times or a dozen times, sometimes only once, but it's often read and expressed multiple times. And as I make multiples of these actin pieces, what I actually get is this long filament. And this filament is 2 monomers per 5.4 nanometers. Okay, remember that nanometer is our scale for roughly 10 atoms, right? So we've got 2 monomers, this long chain here. You can see it's sort of top, bottom, top, bottom, top, bottom, top, bottom. And that is each of these individual pieces here. Now, why do I have these filaments inside of cells? Well, there are a number of reasons, but for one of the ones that we talk about, it is the mobility of these particular cells. Now, I will warn you that this is a word that I have only ever said, or I've only ever read, so I might be saying it wrong, and that word is keratocyte. I think it's a keratocyte. If I'm saying it wrong and it's keratocyte, I don't know. I've only ever read it, but keratocyte is how I'm going to say it, and if that's wrong, I apologize. And what this keratocyte does is when the cell is going to move this way in response to some stimulus or some reasoning or whatever, it builds these cross-hatching actin filaments. And what those cross-hatching actin filaments do is then push the cellular membrane and cause it to basically tip and crawl across the slide, or across whatever surface it's on. This cross-hatching is basically pushing the cellular surface to allow it to move. And that might kind of blow your mind a little bit to go, wait, it's doing what? It's like everyone getting together, everyone in the classroom gets together on one side of the classroom and shoves or pushes with poles or something, basically to try to push over the cell and get it to move. And this locomotion is happening at 0.2 microns per second. And let's be clear, this isn't like the cell says, okay, let's build all the actin filaments and then use them. The cell is expressing the DNA. It is creating mRNA. The mRNA is going to the ribosome. The ribosome is then creating the protein piece, which then gets added to the actin filament on one side of that filament only. And it's not like it just shows up wherever. There is a side that it gets added to, and it gets just tacked on, tacked on, tacked on, tacked on. And this is somewhat, I don't know if it's always, but in many cases, it's a tension-mediated process. And so in this process of reading the DNA, creating the mRNA, translating the mRNA, creating the protein, adding the protein to the filament, we're able to generate a mobility of 0.2 microns per second. Is that fast? Right? Well, compared to what? So you get 0.2 micrometers per second, right? Let's multiply that by 60 seconds in 1 minute. And now we're going to get, 12.0 microns per minute. Is that fast? Well, do you remember your amoeba that you guys did in class the other day that was sort of crawling its way across the slide? What was its speed, right? I don't know that I have that number off the top of my head, but I'm going to look it up to make sure I don't get it wrong. When we calculated our amoeba's speed as it was crawling, we found it to be 7 microns per minute. So this process here is an actin-mediated mobility, which is actually the same kind of thing the amoeba is doing as well. And if you recall, the amoeba had this sort of beginning part in its process where it took a moment to start moving. Part of that is the turn-on process for creating this mRNA to then creating these pieces to then starting this whole process off. Okay. So this paper is fun because it just says, hey, let's make some guesses. Let's see what happens. Let's see what numbers we get. And now we can start to look at how much ATP is required, right? So you can get, 40,000 ATP per second are required for the energy cost of this cell being able to move. And when you start to look at biology at this scale, it just starts to kind of break your head. I guess my most common theme is how does this all work? Right? How does it all keep working? But it does. And you might go, but that's not optimized. That's not the best way to do it. But nature doesn't have to be optimized. Nature doesn't have to do it the best way. Nature just has to do it the good enough way. All right. I hope you're all doing well. I hope you've enjoyed discussing these two papers today. Thank you so much for your time. Have a great day. I'm going to hope that Thursday, tomorrow, you finish up your kinematic model for wound infections. I've decided just to give you that extra day to work on it. And tomorrow, I'm hoping that you're basically putting your presentations together. If for some reason your group isn't there and you're not putting presentations together, not able to do that, I hope that you've reached out to me on Teams. Hopefully I am lucid enough that I've been able to give you some feedback. But we're going to try to, by end of class tomorrow and Thursday, have our presentations, more or less done. And maybe if I'm back in the classroom on May the 4th, like I'm hoping, we can start doing some presentations then. Okay. Thanks a lot. Goodbye for now.